so excited to be here, as Bobby was saying, and thank you for that kind introduction. This is the first time that we've all been together here in La Jolla in person in 3 years. And so, I was thinking about this. xMAP Connect is our premier user group meeting. And so all of you are xMAP experts. And the goal of our time together today is to get together and exchange ideas, talk about our research and our experiences with xMAP technology. So rather than me sit here and try to give you some sort of a technology presentation, I thought it would be more fun if we did something like a journal club. So that's where we're at with xMAP technology, the year in review. So I want to just first, it works, so share a little bit of xMAP statistics with you. So this slide was from probably mid-year, around June, and at that time we had more than 58,000 peer-reviewed publications in the xMAP publications database. And so this is just amazing, because when I joined the company in 1999, there were 5. And I read them in the car on the drive up from Houston to Austin. I wasn't driving, I was riding, for my job interview. So it's a quick read, a couple hours in the car, I can read these papers. But back then, and even in the first few years of the technology, most of the publications were about the technology, because it was new and it was novel. And I always thought to myself that we'll know that we've arrived when the publications are about the science and the research and the discoveries. And xMAP is just one of the tools in the toolbox that was used to achieve that work. And so I think it's safe to say that that's where we are now. Okay. So how did I go about picking the papers that we're going to talk about today? So my methodology. I started with our publications database, and I looked to see all the papers that were retrieved that had been published between January 1st and September 30th of this year. And there were 3,857 papers in total. So, of course, that was too much for me to go through. So then I tried to narrow it down by searching on specific research topics. And I chose these because these are kind of the hot topics that we've been seeing at the conferences that we've been attending in the last year, since things opened up and we could start to meet in person again. So I looked for cancer, cytokines, microbiome, diabetes, infectious diseases, and then COVID-19. I sort of debated about whether to make this a no-COVID talk, but I just couldn't do it because there's so much exciting research being done to try to understand COVID and its impact on us, and a lot of that work is being done with xMAP technology, so it just wouldn't be fair to leave it out. So we're going to have COVID in there too. So after doing all of this, I got down to 72 papers, and then I started going through and trying to pick, what to talk about today. And I ended up with 7 papers that I'm going to present for today's journal club. Now, 7 papers in 30 minutes, obviously we're not going to do a deep dive. We're just going to kind of cover the surface. But what I really wanted to do is try to share with you just the breadth of xMAP technology. So many different areas of research that this technology is being used in, and also the versatility of it. So different levels of plex, different assay formats, and then also different sample types. So we're just going to kind of take a 30,000-foot view of the work that's being published just in this last year. So let's get started. So the first paper is on cancer, and it's paclitaxel liposome for injection, liposu, plus cisplatin versus gemcitabine plus cisplatin, boy, that's a mouthful, in the first-line treatment for locally advanced or metastatic lung squamous cell carcinoma. So liposu is the first commercialized liposomal formulation of paclitaxel, and some early studies have shown promising results. So what they wanted to do was to conduct a more in-depth study, so they did a phase 3 trial. But then kind of on the side, they wanted to look just for the LP group, the liposu group, looking at cytokine expression. So the patients were between 18 and 75 years. They were staged at either locally advanced stage 3B or metastatic stage 4 lung squamous cell carcinoma. And you can see that the two groups were pretty equally distributed. The primary endpoint that they were looking for was progression-free survival. But then they also looked at objective response rates, disease control rate, overall survival, and ultimately the safety profiles of this new drug. Then they wanted to understand the predictive value of plasma cytokines for LP treatment. And so they collected samples plasma at baseline and then at the primary endpoint visit, which was, I think, 15 months after the treatment. And then for this one, they used the Procartiplex 45-plex cytokine panel. And then they looked at the correlation between treatment outcomes and dynamic changes in the levels of the cytokines. So obviously, you can't read this, but this is just a little piece of the data that was published in the paper. And what you're looking at is, in A, you're seeing the cytokine profiles for all of the patients, just all on the same plot. But then in B, you're looking at the partial response. So these are the folks that had a partial response to the treatment. And then on the column on the right, it's these either stable disease or progressive disease. And then you can kind of see from the heat map, they organized the samples by progression-free survival days. So you can see the ones that survived the longest to the left, to the right. So the people in blue, the blue bar represents people that had a partial response. And green is stable disease. And then the progressive disease is, of course, on the end in the red. And then you can kind of see some differences, in the expression patterns. They grouped everything based on the class of cytokine or chemokine and the activity. And so what they found was an analysis of the cytokine levels in the LP group showed that low baseline levels of 27 of the cytokines were associated with an increased objective response rate, so better response to the treatment. 15 cytokines were associated with an improved progression-free survival time. And 14 cytokines, including TNF-alpha, interferon gamma, IL-6, and IL-8, demonstrated an overlapping trend where they have some functionality in common with other cytokines. So in conclusion, they actually found similar results in the LP regimen to the GP group, except that it was associated with lower toxicity, which we would expect because it's liposomal. But then also, they identified a spectrum of different cytokines that could be potentially associated with the clinical benefit of this treatment in patients. So that's obviously a place to do further research. Okay, so next, neonatal chemokine markers to predict subsequent diagnosis of autism and delayed development. So immune dysregulation has been found to be related to a diagnosis of autism. And so there's not a lot of investigations looking at cytokine or chemokine profiles in these groups. So the authors used, in this case, neonatal blood spot specimens to determine patterns of chemokine expression in children either with typical development, who were the controls, 158 of them, delayed development, 69 children, and then autism children, 171. And then they further divided those autism samples into either like low versus high functionality or mild to severe. And they used, in this case, the Bioplex 40-plex chemokine panel. And in addition, they also ran IL-12, P70, and IL-13 to measure the cytokine levels in all of these blood spots. And then they tried to look at associations between certain levels and the specific child diagnosis using various logistic regression and various statistics. Okay, so again, a big chart that we're not going to be able to see, but basically what we're looking at is the rows correspond to the cytokines and chemokines. The spots, the size of the spots correlates to the p-value, and then the color corresponds to the odds ratio. And so each of these, they're comparing the autism to the typical development, the delayed development to the typical development, the autism to the delayed. So they're doing all the pairwise combinations. In the middle, they also did the stratified autism group, and then on the right, some more comparisons. But basically, they observed significant differences in many cytokine chemokine concentrations among the children in these three groups. Exploratory analysis identified CTAC, MIF, and MPIF1 as the strongest predictors of the child's diagnosis. They did not identify any unique predictive markers when they looked at severe versus mild or high versus low performing. And the models that compared the three autism subgroups with the delayed development and typical development produced the same marker selection results. So they at least have a subset of markers that can differentiate autism from the other groups. So high neonatal levels of CTAC were associated with a decreased odds of autism compared to typical development, whereas high levels of MPIF1 were associated with an increased odds of autism, and then MPIF was positively associated with better scores in a number of the different developmental domains that they looked at. So dysregulation of chemokines in early life can impede normal immune and neurobehavioral development. So the study suggests that there are certain peripheral chemokines at birth that may be associated with autism progression during childhood, and that children with autism and delayed development have distinct neonatal chemical profiles that can differentiate their diagnoses. So maybe this is something that can be used, in early life to sort of, keep an eye on these kids and monitor them. Okay, so now we're kind of, we're progressing. We're still looking at cytokines and also related to autism, but in this study, they're looking at zinc as a key regulator of gastrointestinal development, the composition of the microbiome, and the inflammatory response with relevance for autism. So it's known that GI problems and microbiota alterations have been reported in autism spectrum disorders, and then abnormal perinatal trace metal levels have also been found in autism. And then in a mouse model, mice exposed to a zinc deficiency prenatally display features of autism-like behavior. So the authors wanted to model GI development using 3D intestinal organoids grown under zinc-restricted conditions. So in this case, we're basically looking at tissue samples, organoids. So they had control in zinc-deficient organoids, which they processed. And then in this case, the study, they used the multiplexed porcine cytokine chemokine 13-plex panel. And then they used organoid growth media as a negative control. So they wanted to see if low zinc status alone is sufficient to alter GI morphology, so this is done by microscopy, and or trigger pro-inflammatory events analyzed with the cytokine chemokine panel, or whether low zinc status affects microbiota composition, where they did this mostly by real-time PCR and other methods. So again, xMAP is one of the tools in the toolbox that contributed to finding out all of these different things to study this illness. And so here's the data we're interested in, which of course is the cytokines. So they found that organoids grown in low zinc conditions show a trend towards an increase in IL-8, which you can see here, and then a significant increase in IL-18. So IL-8 and IL-18 secretion in the gut epithelium was previously reported to be linked with an inflammatory bowel disease. So it's kind of interesting how these cytokines keep coming up in GI, illnesses. So the results showed that zinc depletion significantly affects the GI organoid development, and zinc deficiency alone could alter signaling pathways that may underlie morphological changes and then promote the inflammatory signaling. They detected abnormalities in a marker protein for tight junction formation and the cellular architecture of the GI tract, not from the cytokine study, but some of the other data that was generated in this case. The microbiota composition of the zinc-deficient mice was also significantly changed. And so maternal zinc deficiency may be an essential contributor to dysbiosis. And then similar changes in microbiota are also seen in autism patients. So in conclusion, abnormal zinc signaling emerges as a central motif linking pathologies in a variety of different places: synaptic pathways, genetic mutations, leaky gut, and increased pro-inflammatory signaling. So prenatal zinc deficiency may therefore share pathomechanisms with maternal immune inactivation. So this is a really interesting study, and something to take further and looking at, picking apart all of these things that trace metal deficiencies can impact children later in life. Okay, moving on. So in this case, we're going to look at a study that was looking at childhood obesity. Multiplex measurements of salivary Phetuin A, insulin, and adiponectin as potential non-invasive biomarkers in childhood obesity. So it's known that obesity increases the risk of developing insulin resistance, diabetes, and cardiovascular disease. So the current study is designed to evaluate the association of these three biomarkers, salivary Phetuin A, insulin, and adiponectin with obesity measures in children. So 76 children were in this study, ages 6 to 10, and they used anthropometric measurements recorded. And then in this case, saliva was the sample being used for the xMAP technology. So based on CDC guidelines, they were able to classify these patients into 3 groups: normal weight, overweight, and obesity. Multiplexed analysis of the 3 salivary markers for Phetuin A, insulin, and adiponectin was performed using assays from R&D systems. And then the diagnostic value of the salivary marker was identified by ROC curve analysis and looking at correlation between obesity measures and then regression analysis. Okay, so here we have just some simple data to look at. So looking at the 3 biomarkers and the differences in the levels measured in the 3 different patient groups. So in panel A, salivary Phetuin A was significantly increased in both the overweight and the obese patients. Salivary insulin levels were significantly increased in the obese and increased but not significantly in the overweight as compared to normal weight. And then in C, adiponectin is highest in the normal weight patients with no significant decline in the overweight patients, but then significantly decreased in the obese. Insulin and adiponectin are significantly different also in the obese compared to just the overweight. So in conclusion, salivary Phetuin A and insulin, are significantly increased in obese children, and adiponectin is decreased. The Phetuin A, insulin, and adiponectin levels in the saliva could be useful to predict future complications such as obesity-related metabolic disease. So the study indicates that children at risk can be recognized by measuring these salivary markers before the onset of clinical and laboratory data of chronic complications. So again, identifying markers that maybe we can use to monitor people in childhood to sort of predict, who we need to be keeping an eye on, who might be at risk for complications later in life. Okay, so now we're going to depart this part of science, and we're going to go into infectious diseases, which, as you may imagine, is kind of my favorite area since I am a microbiologist. Excuse me. And so in this study, they're looking at peptide biomarkers for the diagnosis of dengue infection. So there's an increased population that are at risk of exposure to arthropod-borne flaviviruses. Access to timely and accurate diagnosis could profoundly impact case management. So 20 peptides that were previously identified using a flavivirus proteome microarray were evaluated to determine their discriminatory potential to detect dengue virus infection. So in this case, the investigators developed their own multiplexed xMAP bead-based peptide assay to determine the antibody binding levels to 9 peptides that are recognized by IgM, which they call the PM peptides, and 11 peptides recognized by IgG, the PG peptides. So then they evaluated the assay using a panel of 323 pre-selected human serum samples that were derived from individuals either infected with the 4 dengue serotypes, Zika, yellow fever, chikungunya, West Nile, and HIV, or they received vaccination against yellow fever, tick-borne encephalitis, and Japanese encephalitis virus, and then a set of healthy controls also. So they're basically trying to, kind of get as much data as they can. And so they're looking for the most sensitive assay, but to do this, they targeted a minimum specificity of 80%. So first it had to achieve a specificity of 80%, and then we'll look at the sensitivity. And so here's some of the data. And so basically what they're showing is, and the top is the IgM antibody responses, and the bottom is the IgG, and then in the different groups of samples like the chikungunya, Zika, et cetera, et cetera. And so basically what they found is PG9 had the best sensitivity for acute dengue sera, 73%. FPG1 was the best marker for the convalescent dengue sera with an 86% sensitivity. And then for all acute and convalescent samples, peptides PM22 and FPG1 had the best diagnostic performance with a sensitivity of 60 and 61.1. And the areas under the curve were 0.7865 and 0.8131 respectively. Okay, so they used a random forest algorithm, so basically machine learning, to select the best combination of markers to classify dengue at their targeted specificity of 80% or greater than 80%. And the best model for PM peptides that included both acute and convalescent samples reached a sensitivity of 72.3. For the PG peptides, the best models for acute only, convalescent only, and acute plus convalescent reached sensitivities of 88.9, 89.1, and 88.3 respectively. Antibodies to PG40 was significantly higher in endemic samples versus non-endemic. So they're wondering if this might be a way to differentiate a primary from secondary infection. And then antibodies to PG33 was significantly higher in hospitalized versus non-hospitalized patients. So now they're kind of wondering if that peptide could be used to try to distinguish disease severity. So they found a combination of 6 different peptides for the IgM and IgG models showed improved sensitivity versus sensitivity to single peptides alone. So it's all about, using a large multiplex to kind of look for your hits. And then you can kind of whittle it down with some sort of decision tree analysis to get that best subset. The combination that's going to give you better performance than any single marker on its own, which is the beauty of multiplexing, actually. And they did mention that even though the sensitivity is below 90%, but it's significantly better than anything that they've looked at before. And so this is progress. And so they think that further modification and analysis with larger sample sets from confirmed and undifferentiated fever patients is needed for future test development. Okay, sticking with infectious diseases now, the next study is looking at trying to determine discriminatory antibody fingerprints in active and latent TB. So TB is among the leading causes of death worldwide from an infectious disease. In fact, in the last few years, it's second only to COVID. It was first before COVID. And so we don't have an effective vaccine. So aggressive treatment of active patients may curb spread. And so those of you who have worked in that sort of setting know that you have somebody come in and you suspect TB and they go into isolation until you can confirm that they don't have it. And then we had, when I was in Houston, directly observed therapy to make sure that these people are taking their meds to try to prevent spread across populations. So being able to differentiate active from latent cases though remains a challenge. And so potential differences in both targeted antigens and antibody features may discriminate latent and active TB. So the investigators here assessed the humoral immune response in active and latent TB in patients either with or without HIV to try to identify the most discriminatory markers. So they developed a very large multiplex panel of 209 TB antigens that were coupled to individual magplex microsphere sets. But then for the detection, they didn't just measure, IgG, they measured all of the isotypes and subtypes as well. So pretty comprehensive study. And then they had plasma samples from 4 groups of adults, acute TB with HIV, latent TB with HIV, acute TB, and latent TB and HIV minus. So you can see there's not a lot of patients, 12, 22, 21. So it's not a lot of samples, but it's at least giving them sort of a start to look at this 209-plex that they developed. So here's some data, which we can't see, but you get the idea. I guess one of the things that, as I was putting this together, it's just like trying to pick what data to show you because, it's a lot of data. And when we first started with xMAP technology, before we had the Luminex 100 even, we were running this on a flow cytometer, and you'd get like these reams of data, and then it would take, it'd take you an hour to run the experiment, and then a week to analyze the data. So that's one of the beauties of xMAP technology now with the digital signal processing is you get all the numbers crunched and you get the data right away. But it's still a lot of data to look at. So if you see, you can kind of see some differences on a per antibody antigen basis with that heat map up there. And then, they focused on just some individual peptides or antigens that seem to show some discrimination between the different groups. And so collectively, results from 8 common TB antigens that were used a lot and used before, but then an additional 201 novel antigens, so the 209-plex, provided significant resolution of active and latent TB in HIV-negative populations. Higher levels were found for IgG1 against LAM. Let's see if I can point here. IgG1 against LAM and PPD, and IgG3 for GRO-ES and PPD, and IgA1 against LAM and PPD in active TB cases. And then you can kind of see the distribution here where they're showing the separation of the 4 groups. Differences in antigen recognition were observed in active and latent TB cases that was modulated by the HIV status. So collectively, a set of antigen-specific antibody profiles were defined in both HIV- and HIV-positive populations that provided antigen-specific resolution in TB. Active and latent TB could be discriminated regardless of HIV status based on a combination of antibody levels and Fc binding targeting both well-characterized and some of the novel TB antigens. Active versus latent TB in HIV-positive patients was also resolvable, but this shifted to a profile that included a higher 85A antigen and PST-S3 specific IgM responses. So in latent cases in the HIV-positive, they could get better discrimination by looking at the IgM response. The data reveal new TB-specific immunologic markers that could improve the classification of active versus latent TB. So they concluded that this work identifies a number of novel antibody targets in active and latent TB, so that these are the ones that we can go after to try to guide diagnostic development. They feel like this provides a unique opportunity to build point-of-care diagnostic tools to improve TB medical care. Okay, and now we're into the home stretch. I saved the, best for last, but maybe. So simultaneous measurement of IgM and IgG antibodies to SARS-CoV-2 spike, RBD, and nucleocapsid, multiplexed in a single assay on the xMAP IntelliFlex DRSE flow analyzer. So this paper is very special to us because this group out of Rochester University published the very first paper using the new xMAP IntelliFlex DRSE. And so actually they previously published the method. They had developed this assay on FlexMap 3D and of course would have to use 2 wells to measure IgG and IgM. And so the first paper is the method where they converted this to a dual reporter assay to run on the IntelliFlex. This paper from Microbiology Spectrum is the next step, more of the clinical study, using that tool to look at a longer time course. So they did out to 180 days from the symptom onset. So, just building on that, we built the assay and now let's use it. And so they did this longitudinal study to look at differences in antibody expression over time. So specific antibodies to the spike, receptor binding domain, and nucleocapsid were investigated, 3-plex. They had 310 symptomatic case patients, so more patients now than the previous study that was published. Simultaneous detection of IgM and IgG in the xMAP IntelliFlex DRSE system. And then as another layer onto this, they also looked at the neutralization potential of the antibodies by adding soluble angiotensin converting enzyme 2, ACE2, to block antibody binding. So they used ACE2 as a competitor to block binding and looking for the decrease in signal as an indicator of potential neutralizing activity. And then they did a profile extending out to 180 days from symptom onset, which they described for antibodies specific to each of the antigens in both the IgG and IgM classes. And so you'll see some of this because it's also on a poster, and one of the authors, Steve, is here. And so, we can definitely dive deeper into this study. But basically what you see here is in gray is the IgM and in black is the IgG profile. So this is like over time, days from symptom onset for the S antigen, the RBD, and the N. And then down here, they're looking at for each of, for the RBD, looking at the distribution, if we look at spike, look at two antigens, the spike signal versus N, spike versus RBD, and we can see for IgM and IgG, the distribution of either controls, so unaffected, so obviously they have nothing, so they're down here in the basement, and then the acute and convalescent. And then over here in C, we're looking at the sensitivity, ROC curve analysis for each of the antigens, so S in blue, RBD in red, and N in green. And then down here is the percent area under the curve for each of the days from symptom onset groups. So, and over here is the neutralizing data. So with the ACE, the residual binding, so based on MFI after the addition of the ACE2. So you can see the, differences or lack of differences in the neutralizing potential of each of the antibodies in each IgM and IgG. So put all this together, and the summary is that IgM levels peaked and then declined rapidly at about 3 to 4 weeks post-infection. S and RBD specific IgG plateaued at 80 days from symptom onset. ACE2 more effectively prevented IgM and IgG binding in convalescent cases past 30 days from symptom onset, which suggests greater neutralizing potential. So the xMAP and Teleflex DRSE system enabled simultaneous and semi-quantitative detection of both IgM and IgG at the same time to the 3 different SARS-CoV-2 antigens in a single assay, single well. The assay format is advantageous for rapid and medium throughput using a small volume of specimen. Because remember, you can run 96 well plates, you can run 384 well plates, in an hour or less. Our 384 well is 75 minutes, 96 well plate is 20 minutes. The xMAP and Teleflex DRSE technology demonstrated the potential to include numerous SARS-CoV-2 antigens. So they're thinking about, they could just introduce new bead sets to incorporate different spike variants as new viruses evolve, and still put them all in the single assay. So this could be an important feature for assessing the serological response to emerging variants of SARS-CoV-2, as we move forward in time in this post-COVID pandemic era. So in conclusion, the work highlighted the multiplex and multi-analyte potential of the xMAP and Teleflex DRSE, and provided further evidence for antigen-specific IgM and IgG trajectories in acute and convalescent cases of COVID-19. And so with that, I think we are done. Let's see. Yep, that's all, folks.